Okay, hello everyone. Good afternoon, good morning from me to everyone around the world, because we do have people not just from Southeast Asia, but I know that we've got people in many other countries and regions around the globe. My name is Alison Watson, and it is a pleasure to be moderating this session today as part of the 2022 Biocontrol Technical webinar series of the ASEAN Fall Armyworm Action Plan in collaboration with CSIRO and CABI. We have a very exciting program ahead where we're exploring some new frontiers in the wonderful world of bioprotection. Bioprotection, integrated pest management is a vitally important contribution to global sustainable food production. So not only is this fascinating work, it's also critical to the sustainability of food systems. It is therefore a pleasure to have such a high caliber of speakers and participants joining us throughout this series. I'm just going to move on to the next slide because I really want to just tell you that the session will be recorded and a copy will be posted 1 week after the session. So you will have a copy, a PDF copy of all the presentations and a recording. Today we're going to use the Q&A box to ask questions. So please ask your questions in the end. I'm sure you will have many. In addition, it would be great if you could use the chat box to share thoughts and resources and introduce yourself. And I'm really sorry at the start, I didn't have the chat box going, but it is going now, and I've already got 32 chats in there. So keep that up and keep sharing your thoughts and resources throughout the session. If you'd like to rename yourself, you really just click on participants and you'll see your name. You click on that and you can change your name there by saying more and renaming yourself. Right, this is the third of a very exciting series. We've had already 2 sessions on BT biopesticides, which were fantastic. They are online. The recording for the second session still needs to go online, but that has actually recordings from the field in Thailand of fall armyworm and how to use BT biopesticides to control fall armyworm. So it's a fantastic session. We'll have another session at the end of this August, and following on that, we'll also be looking at nematodes, fungi, semiochemicals, and botanicals. So it's a really a very comprehensive series, and we've got lots of great examples and companies that are working in the field. Please also join our community forum and share your ideas and work. You can do that by going to www.aseanfawaction.org. You click on community, go to the forum, and then you can also ask questions. You can tell us what you're doing, share your work, and share your resources. And it's a great way to build the network. So I really encourage you to do so. Right, today we have a very busy normal. We have 3 wonderful speakers. First up is going to be Dr. Neil Morrison, who leads Oxitec's malaria programs in collaboration with the Bill and Melinda Gates Foundation. Prior roles included leading the Bayer collaboration for fall armyworm and soybean looper and managing overseas agricultural product development collaborations. But recently he has led Oxitec's partnership with Bayer on the development of a self-limiting fall armyworm to support biotech corn and chemistries in Brazil, leading to the deregulation of this product in Brazil. And he's going to be talking to us about that today. Our second speaker is Dr. Arda Örçen. I don't know if I quite got that right, but Arda's going to tell me. He is the co-founder and CTO of Nanomik. Arda is a bio-entrepreneur and interested in biomimicry and biomimetic designs, and has been involved in a number of startups, and will be speaking on biomolecule discovery and microencapsulated biopesticides, delivering biocontrol and encapsulated technology. And our third speaker today is Dr. Damien Fleetwood, who I've seen described as a total fungi nerd by a journalist, which I think is a very high compliment, Damien. Damien is the CEO of Biotelliga, a company mining fungi for cleaner ways to protect crops. Damien has a PhD in molecular genetics and extensive experience in R&D management, and he has been leading several programs on the development of sustainable pesticides and next generation plant protection techniques. So it is wonderful to have three such fantastic speakers join us today. Each one will be loading their own presentation and moving through it, and there will be plenty of time for questions. So please ask your questions to these experts as you want, and we will try and get through them. And I know that they will also try and answer in writing some of your questions as well after they have spoken. So without further ado, I'd like to introduce Neil Morrison. Neil, I'm just going to allow you to share. You should be able to share now, actually. I'm going to stop my share. And I'm going to ask you if you could load your presentation, Neil.
I'm just trying. It says host disabled participant screen sharing.
I'm going to see if I can fix that.
Thank you.
And here we go. That should be, can you share now?
Yeah.
Perfect.
That was easy. Can you see that?
I can indeed, just need to make it in the big screen, and it looks very good.
Okay.
And Neil, it's wonderful to have you join us. So thank you very much.
Oh, thanks so much for the invitation, and thanks everyone for joining. Good morning, good afternoon, good evening, wherever you are. So I'll just kind of set the scene a little bit about who we are and what we set out to do in our mission. We develop new biotechnological solutions for crop destroying and disease spreading pests, including fall armyworms. So there's a massive demand for alternatives to insecticides, as we all know, and we are developing a compelling solution for some of the world's biggest pests, about which I shall tell you in some of these following slides. So just starting a little bit with the technology, and certainly I can, if I don't cover the technology in as much detail as some of the audience like, I can answer questions at the end or dive into the chat. But at this level, with each insect we work with, we insert two genes. One is a marker gene, so that allows us to track and trace our insects and the genes they carry in the environment. And is a super useful monitoring tool to kind of track the success of our males as they do their job out there in the field. The second gene is a self-limiting gene. So that provides maybe two fundamental properties. It allows us to produce males only. So typically it's the males that go out and find mates. That's their typical job in the kind of pest world. And so, we're able to produce these males in a factory setting at very large scale, cost effectively, after release into the field of these male-only cohorts of friendly insects. They find and mate with their pest counterpart females, and at least with the current version of our technology, of the offspring of these matings, the females do not survive. So with successive releases of our friendly males, the number of females in the next generation reduces and the population crashes. So, for example, with our Aedes aegypti mosquito work, we've achieved over 90% pest reduction relative to untreated areas. And I'll talk a little bit more about that particular effort in some of these later slides. Just to kind of finish off on this slide, the two introduced genes that we put into the insect produce proteins that are non-toxic and safe. Whatever eats our insects probably benefit from a good meal, but it's no different to eating a normal pest insect. The technology is self-limiting in the environment. So because it prevents females from surviving, it induces a massive fitness cost on the gene, and the gene rapidly disappears from the environment after we stop releasing. And the fact that this method relies on mating between a male and a female means exquisitely target specific. So, quite the opposite to broad spectrum insecticides in that sense. So just a little bit more detail about the kind of impact we seek to have on a pest population. I mentioned the benefit on the left, which is target pest reduction. The fact that we rely on the mate-seeking behavior of males means that we're able to drive populations to much lower levels than insecticides are typically capable of. Insecticides are very good at knocking down high pest populations to low or moderate levels. But with our technology, we're able to drive pest populations even further because our males are able to find females wherever they are. The second benefit we seek to have on a pest population is to work in tandem with other tools. So the graph on the right shows modeling in which we co-release our friendly male insects with deployment of a BT crop. In the normal way of things, a BT crop eventually succumbs to resistant pests, and you see the grey line in that graph going up. That's insecticide BT resistance essentially going up after a few generations without adequate resistance management. With our insects, we're able to prolong or even extend the durability of that BT trait, and so provide a much more sustainable way of producing crops using BT crops plus friendly males and other IPM techniques. So kind of zooming out from the kind of details of the technology, this image kind of encapsulates what we're seeking to deliver in the sense of a product. So, this shows our Aedes debem, which is a friendly Aedes box. We put our friendly mosquito eggs in a box. We ship them around Brazil, for example, and when they get to the end user, for example, a household, that person just adds water and the box is activated, and friendly males up over the next few days emerge and do their job. So what we're trying to do is move away from a kind of centralized area-wide constraints faced by conventional sterile insect technique, for example. And we're trying to put the technology in the hands of the end user, make it as accessible, flexible, and affordable as possible for governments, communities, and individuals. And just drawing that out a little bit more, it's not just the biotech and the insect that we work on. What we also focus on is the whole suite of tools needed to deliver a cost-effective solution in the field. So all the way from digital tools to enable customer ordering and tracking of products as they move to the field, to packaging a product in such a way that it can move from factory to field using existing distribution networks. Here we show FedEx, just to illustrate the point that what we're trying to do is slot into existing networks to make our products as accessible to growers as they possibly can be. The third picture on that kind of row at the bottom shows a box in a field. So what we're trying to do here, there's nothing more simple than placing a box in a field with friendly insects. This kind of stands in contrast to insecticide sprays, which require manpower, specialized equipment, safety equipment, and can be quite labor intensive to deploy. With our insects, you either just add water or you just place a box down and you rely on the insects to do the rest of the work. So in the case of some pests, a single box could treat hundreds of acres of crop. I won't go through all of these logos in this wall here, but this kind of draws out the point that wherever we work in partnership with local organizations, often global organizations, support our bigger efforts. So, for example, Bayer have partnered with us with fall armyworm and soybean looper. We work with the Bill and Melinda Gates Foundation to develop solutions for important malaria vectors, and the Wellcome Trust support a lot of our work to make our technology more accessible to communities in need. And we work around the world with trusted partners. And lastly, we have an outstanding track record of regulatory approvals and opinions for our technology. So just thinking about what pests we might seek to work on, we might deliver impact against. If the pest mates, if it's relatively amenable to artificial production in the lab and the factory later, and if its life cycle isn't many months long, then our technology is likely feasible against it. So this kind of offers us a universe of targets against which we can apply our technology. And if you look at the kind of range of bugs there at the top of the slide, we're now working outside of insects. Our technology can work against ticks, and if you really stretch it, could feasibly work against vertebrates too. So what this means is that what we are able to do is apply our technology against the world's biggest pests across public health and agriculture. There are thousands of major pests spreading disease and damaging crops. And what we are doing is starting with a fairly limited set. But as you can see from this list, there's some major pests here, fall armyworm being one. So before I kind of go into a bit more depth about our work against fall armyworm, I'll start, if you don't mind, by providing a little bit of context about our work against the dengue spreading mosquito Aedes aegypti. And a lot of that detail is here. So I talked before about our track record of regulatory approvals around the world. Many of these are in the Americas, and many of these, as you can see, are in Brazil. So we are most ahead with our Aedes aegypti work. But you can also see approvals, the kind of green bubbles for field deployments of agricultural pests in the US and in Brazil. And as I said, I'll focus a little bit on Aedes aegypti to start with. So this is the world's most dominant vector of dengue, yellow fever, Zika, chikungunya, and other viral diseases. And it threatens billions of people around the world. And as climate change enables its further spread, it will threaten many billions more. So we have been we've released over 1 billion friendly Aedes aegypti mosquitoes to date around the world, starting with Cayman Islands over a decade ago. A handful of years ago, we transitioned to a new version of our technology, which essentially enables us to deploy, as I described, as eggs in a kind of just add water box. And since we made that transition, we've conducted urban pilots in the city of Indaiatuba in Sao Paulo, demonstrated really the power of the technology to drive mosquito populations down very low levels. And we're now scaling up with the help of the Wellcome Trust, for example, in Brazil, and conducting pilot trials in Florida in the US. anyhow, this kind of shows the timeline of our work with this pest. I'll just kind of highlight some of the key milestones along our kind of journey here. You know, a big one has been deregulation of our Aedes aegypti in Brazil. So what that means is it's fully approved for biosafety and you know, by Brazilian government regulators, and that has enabled us to conduct a beta launch. So, you know, first commercial launch in a part of Brazil in Sao Paulo state. And we did that in the last season, in which we sold our product to businesses, to governments, municipal governments, and also to households who were able to order our product on the product website, and we shipped it to their doorstep within 2 days. So that was a really exciting first step in Brazil, and is essentially the first genetically engineered insect product that has been made commercially available to households on a kind of commercial basis. Now we're looking at scaling up in Brazil. So we're building a very large production and innovation hub in Brazil to expand our capacity. That will allow us to grow in Brazil and the rest of Latin America. And also, as I mentioned, we're making progress in the US. So Florida at the moment, and we're looking to conduct pilots in California next year. All that under the kind of approvals of the Environmental Protection Agency in the US. And this slide kind of gives a flavor of a lot of the marketing materials and kind of product packaging that we've used, with the aim of, you know, what we've tried to do is move away from the insect and provide a really joyful experience for end users, whether that be, you know, pest control service providers or households that are looking to protect their families from biting mosquitoes. And a lot of that has also involved us communicating through, you know, all sorts of channels about our product. And, you know, our beta launch ran in parallel with, you know, an outstandingly positive social media campaign. You know, residents and customers and communities in Sao Paulo state, you know, Brazil's most populous state, welcomed our product with open arms, and it was a really fun, exciting, and challenging at times experience. And just to kind of highlight the openness of you know, Brazilian society to our, you know, quite revolutionary technology. We are selling our product to the second largest bus terminal in the world, Latin America's largest bus terminal in Sao Paulo. Over 90,000 people pass through this terminal every day to travel, you know, around Brazil and the wider region. And, you know, the company that run this terminal, you know, they obviously want to shout about the positive things related to our technology. So, you know, they advertise the fact that they are deploying our product on the premises. So, you know, this was really exciting for us. It's just another kind of affirmation that you know, Brazilian end users are really proud to be using our product on their premises.
3 minutes now.
Okay, thank you. Okay, good timing. Fall armyworm. So similar kind of timeline for fall armyworm. We've been developing this solution since around 2015, partly in partnership with Bayer. And, you know, that led to us conducting the first pilot trials in Brazil around about 2019, 2021. In 2020, just like with our friendly Aedes, we were fully deregulated by Brazilian regulators. So, in effect, ready to sell in Brazil. But first, what we want to do is build the kind of deployment muscle to enable us to deliver impact at scale. So, since 2021, we've been conducting farm scale pilots over thousands of acres of corn in some of the biggest corn growing regions in Brazil. And so, looking ahead, this is a massive stepping stone for us learning about how our product will fit into farmers' lives, deliver benefits to growers, and also protect the biotech corn traits that Brazilian corn production so heavily relies on. And as we start to kind of roll out, we hope across millions of acres of corn in Brazil, our aim is to improve the sustainability of Brazilian corn production overall. Okay, some modeling, which I'll run through very quickly. But essentially, this slide speaks to those two benefits that our technology provides. Looking at the kind of top row of panels, this describes a scenario in which corn carries one Bt protein, and in which case releases of our friendly fall armyworm over that Bt crop are able to extend the durability of the Bt crop in the face of resistant fall armyworm for many, many generations. But on the right, you can see population size is kept low when we release at moderate levels. Looking at the panel below, where corn carries multiple corn traits, they work much better together. So what we're able to do is essentially protect the durability of double traded corn products for perpetuity, or at least over the very long term, and in parallel also keep fall armyworm population sizes to very low levels. So I guess two simple takeaway messages here are: as a standalone product, friendly fall armyworm can provide a really valuable biological control benefit. But used in combination with Bt crops, and maybe also insecticides, they can work together extremely well to provide a kind of synergistic effect in managing fall armyworm over the very long term. We're building a factory in Brazil. Again, as we start to kind of ramp up our mosquito efforts, we are doing the same for fall armyworm in preparation for really large-scale impact in Brazil. And I'll just end with this, which kind of speaks to what we need to do to really deliver impact for global food production sustainability. So, this kind of speaks to all the points I've talked about beyond the kind of biotech detail, delivering scalable and cost-effective manufacturing. So, flexible and robust distribution, simple deployment that really fits with farmers' current activities, and makes it really easy for them to kind of use our product on their crops. As I've kind of spoken about, it fits really well with integrated pest management strategies. So it's biological, it's environmentally friendly, but it also is able to enhance the durability of other products used alongside it. It's biological.
Thanks, Neil. I'm just going to stop you there because we've got a lot of questions.
Okay.
And we're running out of time a little bit. So we're going to go through these quite fast paced. So you ready? It's sort of like a bit of a, one of those things on TV where you get fired questions. But here we go. Lots of great questions. Here's one. Great presentation. Thanks for sharing. What are the prospects for the Southeast Asia market? I can imagine some regulatory challenges. Any pilots ongoing as of yet? And if so, what products and targets and where?
Not yet. So our focus has been on Brazil. And it still is to some extent. However, we are now starting to kind of raise our focus beyond Brazil, looking at Africa, Asia, Australasia as potential markets. I can't really offer any more insight than that, only from the, for the reason that we're so early in thinking it through that no immediate plans. Obviously, the regulatory pathways will be different in different countries. I think the progress we made in Brazil as a really positive precedent in a country with robust regulatory frameworks can only help in that sense.
Yep, for sure. That's excellent. And it's good to have, I mean, you'll find out a lot, I guess, around how it works and how best to communicate with farmers through that process. So that's going to be very valuable when you move into other regions. Here's another question here for the friendly product. What is the optimum plot size or field size for demonstration?
It kind of depends. Okay, it depends what we are seeking to do. I mean, at the moment, our aim is to understand how the product behaves in a typical corn growing landscape. So what we've been doing is deploying over thousands of acres of corn in Brazilian corn. That's the kind of scale that they do things in Brazil, obviously. And that has allowed us to track the movement and other kind of behavioral factors of the moths that we deploy. In terms of providing a suppression effect, because fall armyworm moves reasonable distances, what we're doing is seeking to understand how a single customer could benefit from deploying our solution, and at least from this early stage, perhaps at the kind of farm scale, we could potentially deliver a single customer solution. When it comes to friendly Aedes aegypti, they move much less far, maybe 50 meters in their lifetime. So the kind of focus of population suppression benefit we can have is much kind of more focused, if that makes sense.
Yep, no, that does make sense. It's a good point.
Okay.
Here's a good question here. The product produces good results. It's a great invention. I'm interested, however, to know, how does it affect the agroecology? Because in nature, there's multiple pests, and such suppression of one population may promote another problem for another species. What is your view on this?
I think typically what we're trying to do is step into fill gaps that existing kind of pest management strategies are failing to kind of fill. So fall armyworm is an obvious one. It's a standout pest in Brazil, for example, because it develops resistance to insecticides and other tools. What we're doing is stepping in to fill that gap, and hopefully, off the back of that, farmers will be able to, produce crops, with far fewer inputs like sprayed insecticides.
Yeah. And are you doing trials with biopesticides? I mean, you mentioned sort of more conventional pesticides and BT crops as well, but are you doing trials in conjunction with biopesticides, bioprotection?
Not yet, but part of our thinking over the next year or so will be, how do we kind of deploy alongside sprayed insecticides, whether they be synthetic or biological.
Yeah. Okay, thanks, Neil. One last question, then I'm going to move on to the next speaker, but you have a lot of questions in the Q&A box, Neil, so you may be a bit busy, but there's a question here: will the pests be able to develop resistance over time?
I mean, I'd start by saying, no pest management solution is 100% immune from resistance developing. So, we couldn't claim that. What we've never seen is resistance developing after, over 1 billion friendly male mosquitoes released. And also, just to kind of finish on that, our insects dilute resistance to BT crops and insecticides, but actually they have the same effect on resistance to the friendly approach itself, so that were resistance to develop against friendly insects by some mechanism, friendly insects can also dilute that resistance and control resistance to itself.
Okay, excellent. Hey, Neil, thank you so much for joining us. There's lots of questions there. I think you may be a bit busy, but look, it was fantastic to have you introduce this, and it's something that we haven't talked about in the series to date, and we've had a very extensive series. So it's really exciting to have you join us and talk about this, and I'm sure we're going to hear more. We'll invite you back in a year's time to hear how the trials are progressing in Brazil and your plans for the future, and hopefully where that may intersect as well with Southeast Asia in our region. So we'll keep in touch. Thank you, Neil. And if you can stay with us, that would be great. I'm now just going to move quickly on to our next speaker, Arda, who I've already introduced. Arda, if you could share your screen now, that would be fantastic. And we're very much looking forward to a discussion around encapsulated biopesticide technology. Very excited to hear about this. So welcome. And if you can start, that would be great. And I can see your screen perfectly.
Okay. Thank you, Alison. Hello, everybody. I'm Arda, co-founder and CTO of Nanomik Biotechnology. As Nanomik Biotechnology, we are producing next generation microencapsulated biopesticides for the agriculture, sustainable agriculture. So before the Nanomik, I'm also a bio-entrepreneur, and I have a background of the molecular biology, and also I'm doing my PhD about the developing of the protein-based biopesticides. And Nanomik also founded in 2015 by co-founders, me and my co-founder, and up to now we closed 60K pre-seed investment, and we have investors from Denmark, Netherlands, and Turkey. We are developing a baseline technology, microencapsulation baseline technology. But in the meantime, we are also developing new biomolecule discovery systems, and we have products for pre-harvest usage and post-harvest usage. And up to now, we also collaborated and awarded from different kind of companies, and also we are part of the EIT Food and EIT Climate, and also we are we have prize from the Biomimicry Institute from United States. So before the technology, what is the idea to start this business for us? The main idea is that we inspired from the plants, because plants have able to produce volatile molecules, and these molecules can act as a everybody knows that as a activity against fungi, insects, or herbivores. But in the meantime, these molecules can able to have specialty to warn other plants in the neighbor to protect themselves against these pathogens in time by time. So of course, in the industry, in the product development industry, there are lots of products which comes from the specific herbs and plants and used as a botanicals against pathogens. But the general problem of this type of biopesticides are their stability. They have a stability problem, persistent problem, and they have to be used multiple spraying times. So this caused low price performance, and they don't show stable activity season by season. So this is the general problem of this type of botanical molecules. So because actually why they couldn't able to show standard activity in season by season, because they directly affected from the physical and chemical factors in the environment. So these volatile molecules can be sensitive against UV degradation, oxidation, and also heat degradation, and they lost the activity. Then they couldn't have enough time to act as a biopesticide in the on the plants. So beyond these problems, we as a company, we want to understand their combinatorial synergistic activity against specific pathogens, plant pathogens, and we started to understand this whole biomolecule, plant-based volatile biomolecule library, and we tested their combinatorial effect in our high throughput screening systems. So, and we found lots of specific combinations in the whole plant library, plant molecule, volatile molecule library, and then we tested again and again. But we understand that these molecules have to be protected when they sprayed to the crops. So then we developed our microencapsulation microsome technology. So what are we doing? We are the specific volatile molecules we are putting in a small microcapsules, and we are gaining them as a controlled release mechanisms in different kind of chemical and physical trigger conditions. So this technology also patented, and we have Deep Tech Award from Climate Launchpad. So the advantage of our technology is it's not specific for the plant secondary metabolites or volatile metabolite. It can also be used for the antimicrobial peptides or proteins or specific capsule proteins. So these capsules, when they apply to the crops, these capsules control the release in different pH conditions or the water volume. So, and it gives longer shelf life to the molecules, and also it's very suitable for all different pesticidal application, fungicidal or insecticidal. And when we compare with the traditional encapsulation technology, our technology allows a high rate encapsulation efficacy, and the machinery also easy to scale up. So the capsules which we generated, the diameters almost up to 5 micrometers, and the frequency is so narrow, and it's the specific size of these capsules directly affects on the pesticidal activity, because the similar size of the capsules can able to penetrate plant tissue in the same time. So in our studies, we found that in different pH conditions, we tested in different pH conditions. In acidic pH, our capsules release more active ingredients when compared to the neutral pH. So these capsules it gains the tunability effect. So when you use the pH conditioner in the spraying tank, you can it depends to the conditions, disease rate in the field. You can use as a preventive, also curative product. And also in neutral pH, capsules are more closer to the zero order kinetics. So the water content is also important. When the water content in the microenvironment of the tissue of plant is high, the capsules going to be swell. It's getting bigger, and the porosity on the surface of the capsules it's increased, and the active molecules can able to diffuse outside. So our capsules directly affect on the loss of cell wall integrity. So when the capsules attach to the surface of the fungi, so it's start to control release, and the biomolecules which already we used have an effect on the membrane, and it start to disrupt and also start the ion leakage from inside to the outside of the cells. So when we compare with the traditional botanical formulations, actually they have poor adhesion and uptake problem on the surface and in the foliar applications, and also the penetration problem in the soil applications. So our technology also helps the extended adhesion and uptake of the plant tissues in foliar application, and we can also protect our biomolecules for the soil-based applications. So in the playground of our technology, we of course started from the developing technology, but we choose crop protection area and also food preservation because of the revolution of the biological molecules occurs in this industries. So in crop protection area, up to now, we have biofungicides, and also we are developing our bioinsecticides. And next year we plan to launch our bionematicide products. Our first product is the Mikox. Mikox is the generated from our baseline technology, and it's cost-effective, standardized efficacy, and residue-free, and it contains thymol, geraniol, and oregano oil, and it's directly have an effect on the specific plant pathogen fungi, and be approved in different plants. So when we compare with the activity in the field with the traditional botanical pesticide, so our technology and our products almost 30% higher than traditional applications, and we can we can catch the synthetic fungicide application period almost. So we tested our products with different partners via Crop Science and BSF, and also Hektaş from Turkey. So we have specific tests, field studies for the benchmarking test. Actually, we tested against with the regalia. It's the one of the important fungicide and biofungicide on the market, and we also understand the active standalone usage. So application interval almost 10 to 12 days, and the dosage rate of the regalia, the active dosage rate is 200 milliliter. So when we compare with the activity in the field, we catch the 70% disease prevention rate when we compare with the active dosage of the regalia. So when we increase the dosage of our product, we catch the 90% disease prevention. So we also want to understand the comparative benchmark test with the biopesticide, different biopesticide. The Serenade is one of the important products in Turkey, which we used, generally used. It's the bacillus-based active ingredients, and almost they generally used 1.5 liters for 100 liter dosage. So we can catch the specific results, and almost same activity is 300 milliliter per 100 liter dosage, and it means the minimum 70% disease prevention rate. So our active dosage we offered to our customers is 500 milliliter per 100 liter dosage, and in this test, the disease rate was recorded 40% botrytis. So we also want to understand in the spraying program, the behavior of our product in the spraying program. So we compared with the the Bayer crop science program, and we integrate our products after the flowering period. And we tested against a powder mildew. And we also, in one line, we as a single window, we want to use from flowering up to the harvest.
2 minutes. 2 minutes, Arda.
Sorry. So this in this test, we saw that Micox, when Micox used in the spraying program, the synthetic fungicide spraying program, after the flowering, catch the same efficacy of the fully synthetic program. So it means that we have very collaborative activity in the synthetic program. So it's very easy to use. The microcapsules I mentioned before, when they use to the products, crops, it's start to slow release and protect the fruits or the green body parts. So we have also post-harvest effect. Our products have an activity on the quality of the product. Also not for the vineyard application, for strawberry, and also for greenhouse applications. And with this technology also we produced a bio post-harvest product. And with this product, we almost increased the shelf life of the nectarines and also peach almost 2 times higher. And we have, when we compare with the traditional usage, we have almost 50% higher activity when we compare with the traditional applications. And for grapes and citrus products. So our we are also preparing our bioinsecticide. It's called Talos, and it contains orange oil and neem oil, and also generated with our technology. And we have proven effect on the tuta absoluta in from larva 2 to larva 4, and we catch the higher kill rate in after 2 to 3 days in the greenhouse conditions. Also have an effect on the acars and the egg laying reduction on the acars to spotted spider mites actually. And we have a significant impact on the climate, and we can help to eliminate 65 tons of carbon dioxide emission when they compare with the chemical and synthetic fungicide-based applications. So we have value propositions. We have residue-free, multi-effective, stable, and user-friendly product portfolio. And also our discovery, we also have a workflow for the discovering of the biomolecules, and we are investigating biomolecules from the fungi, bacteria, and plants. And we have we found specific peptides, antifungal peptides from the fungi, specifically entomopathogenic fungi. And we have identified more than 100 peptides, which directly kills the fungi and specifically for fungi, and no effect on the mammalian or cells or bacteria. And it's a very in the nanomolar and degree activity against specific pathogens. And it has its induced reactive oxygen production in the fungi and also regulated cell death. So we are preparing these products against specifically for oomycetes and also necrotrophic fungi. So we develop a recombinant-based expression system on yeast, because these peptides are cysteine-rich residue, have cysteine-rich residues and don't it can be produced by bacteria or specific algae. They very suitable for the yeast-based expression systems. and we hope to reduce, targeting to reduce this peptides production from to actually up to 0.5 to 1 euro per gram with our manufacturing technology. Thank you for listening to me.
Thank you so much for that presentation. There's lots of information there. And just to remind everyone, we will be sharing all the presentations. So you will be able to look through Arda's presentation in full. Lots of interesting stuff. It's extremely interesting. I also like how you looked at the climate impact as well, which is really important. Other sort of plant pests and diseases that are really important in this region were mentioned. I heard Tuta absoluta as well, which is something of interest to many in the room. Quite a few questions here for you, and we'll be quite quick, Arda, but here's a question around the waste. These I guess this new technology, is it biodegradable, or how does it end up in the environment?
Yeah, actually, our product, our microcapsule, our microcapsules generated from the biopolymers. So this biopolymer is fully degradable in environment, and also it's safe for the consumption for the mammalians and also humans.
Yeah.
Yeah, it's generated from the blend of the fungi also.
Oh, excellent. When you apply it, does temperature or weather have an impact on the efficacy of the microcapsules? I mean, if it rains, or if it's too hot, or if it's too cold, I mean, does that have an impact on how the microcapsule breaks down or lets the product work?
So in hard conditions, of course, in the fully rainy conditions, if I understand the question true, but in the hardest conditions, the capsules also, of course, you have to spray, repeat your spray, of course, but it's the same as in all different biopesticides. Yep. But it's more, I can say it's more resistant to the specific conditions because the adhesivity is so higher than its adhesivity is so higher. I mean that to the plant green body parts.
Excellent. Okay, thanks. That's a good answer. And here's a question here from Cyril. Hello, I'm very interested on this product. Are they available here in the Asia markets yet, specifically for Philippines? I would like to use it with entomotic pathogens in the future. Where are you currently using this or trialing it? In which countries and regions, and what's your sort of interest in the future?
Actually, now we are currently in Turkey. In the next years, we want to extend our market actually to the Africa and also to the Asia. So, but now we are currently testing our products in the countries in nearby in the Mediterranean side as a testing. After the all testing results in different climate conditions, of course, we want to be in the market.
Yep. Excellent. And here's a question here. How effective could it be to protect maize grains against contamination by mycotoxins?
So we have a specific tests in with the company who is one of the biggest company in the world. We have a collaboration now, and we are testing our products in a different stage of the mycotoxin production. From we are going to start for the soil application against Fusarium for the maize, and then we are going to test our microencapsulation technology if suitable or not in the silo-based application. So we are want to understand from the field to the silo the full we hope the full protection system, and now we are testing our products in there.
Great. Well, it sounds like you've got a busy time ahead of you, Arda, and it's really interesting to see this, and it looks it looks actually fantastic work. Looking forward to hearing more about it within the next year as well. We'll invite you back as well to see how progress is going, and hopefully I can see lots of people within this region interested to work with you as well, to see if they can maybe partner. So you've got a few questions there in the Q&A. So thank you so much for joining us. Absolutely fantastic to have you. So thanks, Arda. Thank you very much. And that brings us to our last presenter today, but no means least, and it's really fantastic to have Damien Fleetwood join us from New Zealand. Damien, welcome. If you could share your screen, which you're already doing, he's onto it, and we're going to hear now about your work, which is also very exciting. So I see there's some chats that may have some relevance to your work as well in the chat box from Carol, etc. So please join us, and please go ahead.
Cool. Yeah, thanks very much. Thanks. Really appreciate the invitation to talk to you all around the world this evening. Tough acts to follow from Arda and Neil, but I'll do my best. Now, Alison, feel free to cut me off. I'm actually going to present this a little differently than I'd planned. So if I go over, just cut me out. No problem. Now, my name's Alison said, Damien. I'm the CEO of Biotelliga, biological crop protection technology company based where I am right now here in Auckland, New Zealand. It's 9:06 p.m. in the evening here. This evening, I'm going to give you a kind of a speed tour through just a couple of the technologies that we're developing here that are based, I guess, most relevant for fall armyworm, certainly involved in insect control, although to tell you right up front, we haven't yet done much or any work on fall armyworm directly. It's something that we're certainly interested in. Now, just a little bit of background about the company and what we're trying to do. Essentially, we're attempting to develop biological crop protection technology that can really work in the way that growers might, at the moment, use synthetic chemistry. And I guess, Arda pointed out some of the kind of problems or deficiencies existing in biologicals as they have been to date. And I think we see the issues with biologicals that need solving as principally being around efficacy. Certainly high level efficacy is desirable, but even more than that, I think consistency is really important. So a lot of existing biologicals, certainly kind of traditional biocontrol technologies, will work quite well one day, moderately the next, and not at all the next. And growers really need more consistency than that going forward in kind of next generation biologicals. Obviously, cost effectiveness is another thing that has plagued biologicals in their introduction into wide-scale use in agriculture. So in this company, we're a company of biologists, really, and to try and solve those issues, we really come at it from understanding the biology of the systems we worked at. And as Alison said, we're pretty well, all of us, especially me, but really fungus lovers here, and the way that plants and fungi interact. And we mine those interactions to identify and develop technologies. And principally that means for certainly biopesticide development, which is the main part of what I'm about to talk to you about. It means understanding what the biomolecules are that are involved in complex biological extracts that are responsible for efficacy, such that we can QC products that they always have the same amount of those biomolecules, so that they will be highly efficacious, particularly, as I mentioned, consistent in their activity. The technologies we're developing in biopesticides are process neutral. As Neil pointed out, it's really important that growers can use things in a way that's easy and that they're used to doing. So take out chemistry, put in biologicals in this case. Shelf life stable, and stability is another general issue for biologicals. And as I mentioned, obviously, technologies need to be cost effective. So this is kind of, broadly speaking, the 4 technology areas or the active substance areas that we're developing at Biotelliga in our pipeline. And 3 out of these 4 are biomolecule-based biopesticides, so sprayable biopesticides to use. And we won't have time this evening to talk about the phytotoxics that we're developing as bioherbicides. Neither do we have very much time to talk about our biofungicide kind of flagship technology, which is quite close to market. But I would just point out, given our focus on Southeast Asia on the call this evening, that one of the principal diseases that our biofungicide in development is active against is rice blast. This is from a field trial in the Philippines, and you can see that application of the code name SS7.9 biofungicide is very effective at controlling blasts. So if anyone's interested, maybe whether it's in the chat or in a follow-up email, we'd be happy to talk about that technology further. But what I am going to talk to you more about this evening is principally NZ7000, code for bioinsecticide in development. Pre-commercial, we expect registration here in New Zealand next year. And I'm going to talk to you about what we call Endogen, our platform area for identifying fungal endophytes, so fungi that live within plants for the life of the plant, delivering a given bioprotection trait all the way through from planting through to harvest. So I'll start with showing you one of my favorite pictures. So this is the biological system that we mine for our bioinsecticide technology. Now, this is very familiar to people in New Zealand. I see there are a few people from here on the call this evening, maybe less so in some other parts of the world. This is a grass field, as you can see. It's a forage, cool season grass. It's called perennial ryegrass. It's one of the principal forage grasses growing here in New Zealand for animal forage. And this field here on the left is exactly the same planted grass as this one on the right. Neither of these fields have been treated any differently. They're not irrigated differently. No pesticides have been applied. But this grass on the right has been completely eaten by insect pests. This one on the left hasn't. It's exactly the same grass, exactly the same cultivar of grass. The difference between these fields is that this field on the left has been infected with a fungal endophyte, endo inside plant, phyte plant, fungi that living within all of the tissues of this grass plant. As you can see here in blue, hyper in a longitudinal section of a leaf, and each of these green dots is a GFP genetically labeled hyper inside a cross section through a grass tiller. So you can see that this field here is very widely infected with a fungus. And the reason the presence of this fungus leads to really extraordinary insect control, and you can see the black and white difference between these fields, is the production of biomolecules. And in this case, another field showing something very similar, in this case, a meadow fescue plant. It's been infected with this endophytic fungus called Epicloe, in this case, Epicloe uncinata. It's infecting these grasses, and it's producing these molecules called lolines. It's a pyrrolizidine with this kind of funky oxygen ring and various decorated amine. And it's the production of these biomolecules that are very insecticidal that lead to this really significant biocontrol impact. I think it's probably the most significant biocontrol naturally occurring, depending on if you count BT, which I'm not for the purposes of this evening. Obviously, the impact in this setting is really substantial, but it's very low value as a plant. And the technology we wish to develop was to utilize this really high level naturally occurring insecticidal phenomenon to develop a biopesticide spray utilizing these molecules in the system to spray on food crops. And indeed, that's what we've done. And I'll tell you more about that in the next 5 minutes or so. So the path to doing that is we've developed a system of taking the fungus that naturally and obligately lives within this grass plant and protects it naturally. We take the fungus out and we're able to grow that accidentally using various tricks. And further to that, using even more tricks in bioreactors, we're able to induce it to produce these molecules in large scale reactors. And that's a very short version of a very long story, which leads to development of this biopesticide spray. Actually, the product will be called endophyte, Endo-F-I-G-H-T. Now, the features, as I've mentioned, this will be a contact, or is a contact foliar insecticide product. The active substance is a fermentation extract of this fungus Epicloe uncinata, a proprietary strain of that. And we QC, as I said earlier on, the product such that it contains known amounts of these marker compounds, lolines, and there's a mixture of these, which is essentially what leads to the insecticidal activity. This product is very active against, and I'll show you some quantified data in the next few slides, against a range of insect pests. We've mostly focused on sucking insects, particularly aphids, thrips, and whitefly. Our first label will be for mealybug in wine and grapes, but a range of others as well. Some evidence for fall armyworm, although we haven't explored that ourselves just yet. We'll be targeting a range of segments, including high volume row crop segments, which is reasonably unusual for a biological. The reason that we are able to do that is because over the last couple of years, we've been actually really successful in genetically engineering very fast growing reactor tractable organisms, like a brewing organism, to heterologously express the genes for this molecule's production. So we take the genes from the naturally occurring endophytic fungus, we engineer those into bioreactor tractable organisms, which enable us to produce very low cost fermentation systems. We're not quite there yet, but we're most of the way, which is pretty exciting for us. We have a stable liquid formulation. Importantly, obviously in this industry, there are no toxicological endpoints of concern, either for us or for honeybees and other beneficial insects that we've trialed either. And I think a really important point about biologicals in terms of benefits compared to synthetic chemistry is that this product is readily biodegradable, will go away in the environment. And it's particularly important if there are unforeseen circumstances, as there have been, as we know, with a range of chemical products. And useful for use all the way through to harvest. The New Zealand regulators indicate no issues with residues or withholding period, and we expect that to be the case everywhere else in the world also. Now, just to quantify some of that activity, I think probably the audience is principally interested in what it works on and how well. NZ7000's lowlands containing extract of Epichloroides and Arda fermentation have insecticidal activity demonstrated by us or previously in the literature against a range of things. I mentioned mostly we're focused on sucking insects. An example of that's shown here in our laboratory, using the product on green peach aphid on bok choy, actually. This is the control. This is the percentage of either mortality or incapacitation over 24, 48, and 72 hours. The light bars are essentially stopping of feeding. The dark bars are mortality. And you can see that over that 3-day period, pretty much complete control of green peach aphids in the system, very much analogous to the comparator, which is one of the leading chemistries, obviously, in imidacloprid, clearly being removed from various markets at the moment. Diamondback moth is something that we've shown efficacy for recently, so we do think that there's some likelihood of lepidopteran control. Now, there are multiple modes of action, as well as being insecticidal. This product has repellence activity. I'm going to race through this. You can see the effect here on a different aphid species, in this case on barley. And given the choice, aphids will very rapidly migrate from plants that have been sprayed with NZ7000 onto plants that have been sprayed with water. That activity goes out well past 8 days, past 2 weeks. We're not yet sure quite how long it lasts. This has been noted for other things by us and in the literature as well, including some years ago now by Riedel and their colleagues in fall armyworm. And obviously we've performed a range of field trials. This is an example of a regulatory trial in wine grapes with mealybug control, and 14 days post-application or at harvest time, some 3 weeks later, compared to incidence on the untreated control, we get very significant reduction at the field rate in half the field rate. And this is comparing to spirotetramat. A range of other, you can read them for yourself here, insects controlled on a range of other plant species. This has been trialed by us and a range of partners around the world. We're really confident in the very high level efficacy of this product. It's not magical, and there are a few things we've trialed it against in which it doesn't work, but it is a very broad spectrum, highly potent, very consistent biological insecticide, and we're really very excited to bring it to market relatively soon. We expect our first registration here in New Zealand in 2023, next year. We've just about completed the final tick off on the New Zealand EPA dossier, and we will be looking to register around the world after that. We're talking to the US EPA right now. So I want to move quickly spend a little bit less time, just come back to this idea of endophytes. So just to remind you, this is coming back to the biological system that we've developed in NZ7000 as a biopesticide spray from, but we kind of stepped back and thought, well, no one ever needs to spray this particular plant, and what about it's got this endophyte living within it that's protecting it from within all the way through the life of the crop? what if we could utilize this kind of system for crop protection in wheat or in corn or in rice, indeed any other crop? And that's a platform we've developed that we are calling Endogen as a way to deliver biocontrol traits into modern crops using these endophytic fungi that live within plants. So the very short version of oops, is it going to come up? Yep. Of a pretty long story now is, we had a big idea and we went out and tested it. And I think the critical thing to understand, to the background that we come at this from, is that it seems pretty clear, and I think it is clear, that modern crops don't really have any selection pressure to cultivate or adapt to symbionts with microbes that are beneficial. So modern crops essentially lost this, many of the bioprotective endophytes. They still have many microbes living within them, but there's not been that pressure to maintain bioprotective relationships during domestication, certainly over the last 100 years, but much longer than that too. If you compare a modern crop, which is covered in pesticides and is helped with fertilizer and irrigation, compared to where these wild ancestral plants to these grow, you can see that there's a lot more pressure to work with microbes and living within them to have these bioprotective relationships. So we went out and we have indeed gone to the parts of the world where these ancestral crops are from. And we've brought back from their material back to New Zealand, from the ancestral plants to modern crops. We've isolated thousands of endophytes, endophytic fungi from within those plants. We've then asked the question, can we deliver those endophytes, which have come from ancestral plants, first into modern crop species? I'm using wheat in the next few slides as an example for that, but we also do this with corn. Do those endophytes, are they able to infect modern crop derivatives like modern wheat plants? Further, do they persist within, thank you, within that wheat plant for the life of the crop all the way through to harvest? And once we understand that, we then ask the question of those endophytes, do they have any biocontrol traits? And we'd expect that they would, given a close relationship such as that. So this kind of shows an example of taking one of those ancestral endophytes, a wild endophyte if you like, applying it to a modern plant, in this case wheat, and then determining whether those plants all the way through to maturity maintain a systemic fungal infection. And the answer, and many more than I wildly dreamed, is yes. So we have a very large number of endophytes in our collection that have come from wild ancestral species that we can systemically, persistently inoculate onto a seed that will live within modern wheat plant for the life of the wheat. And this is also true of corn. This is the last slide. We've been just now in the past year, so this is certainly pre-field work. This is all work in the lab, and the next year we will be taking some of these out into the field for development. We've started to then ask the question, we've got a really large number now of endophytes in our collection that we can infect into modern crops and will persist within them. What are the biocontrol traits that might be present? And we've looked at, this is an example with Rhizoctonia, a big establishment disease issue in a range of plants. And this is just showing you the effect that one of the endophytes has. It's basically complete control of Rhizoctonia disease and a population of wheat plants shown graphically here, but you don't really need to see the graph. And we have a range of bioprotective disease controlling endophytes of wheat. We've tested in vitro a range of other these, and Fusarium pythium and a range of others, in fact, as well. And there's a lot of bioactivity. We've just now started to move into insects and developed an aphid repellent screen. And believe it or not, the very first endophyte we put through that screen showed really quite significant repellents for green peach aphids. So we're really excited to develop new insect screens as well. And to remind you, this is the main thing that we see in terms of bioprotection from our exemplar endophyte in the grass system. So we really are keen to explore insect resistance quite a lot more. And as anyone that's familiar with this area probably aware, we also see a very large number of biostimulant, in our case, water deficit, salinity, heat stress protection from the endophyte collection. So we're excited to take this to the next stage now. We'd be very interested in looking at fall armyworm if there are any partners out there that are skilled in doing so. We anticipate taking our first products. We've got lots of validated candidates now from whole plants into market around 2026. So just how this might pertain to fall armyworm, Arda, As I've mentioned, we've developed the NZ7000 bioinsecticide based on a endophyte-produced metabolite principally. It works really well. We don't yet know around Spodoptera. As I mentioned, we're very happy to partner if there are interested parties at looking at this. I've just mentioned we've got a large number of endophytes I was focused on. We also have a large number in maize. We've really focused on disease control, but as I mentioned, we will be moving into insect control screening very, very soon. And I can see that this could be really disruptive in terms of the way that this technology could be applied in the general IPM program going forward. As I've mentioned a number of times now, we'd be really delighted to explore any partnership potential in the Southeast Asian region. And I'll just close with maybe many of you probably unaware that fall armyworm has recently been found in a large number of places here in New Zealand. It's not been found previously. So broadly found now in the North Island, and we're nervously waiting for colder days through the winter, hoping that there will be no populations survive. But unfortunately, we are having rather a warm winter, so some concern we may have fall armyworm problems in New Zealand coming in the spring. Thank you very much indeed. It's really nice to talk to you, and happy to take some questions.
Thanks, Damien. That's excellent. I'm just going to tell everyone we're going to be 5 minutes late ending, just because I want to keep 5 minutes to ask some questions to Damien, and then I'll wrap up very quickly. But fantastic presentation, really interesting. It's sort of almost like unlocking the power of endophytes. So nice to have that sort of to look forward to, because it looks like such a exciting and really transformative area of research for plant protection and health. So thanks, Damien. A couple of questions here, and we've got one right here. Do the endophytes increase the immunity of the plant to fight the pest, or the endophytes directly fight the pest, or both? And another question is, do they kill them through a toxin, or do they repel them?
Yeah, a combination. So, you know, we haven't got to grips with mode of action for a lot of the things we're doing. We've worked through really large numbers of screens and we've validated those. We're just moving into a point now where we're understanding how things work. I can say it definitely is both. So there are a range of ways in which these organisms can work, you know, niche occupation, antagonism, competition, and biomolecule production. I can say that there is quite a lot of biomolecule production and direct activity that way, but we also anticipate there'll be quite an impact on the plant and stimulating the plant effect that's yet to be explored, but we know it'll be all of those things. I'm sorry, I missed the second part of the question, Alison.
The next question was, do they repel them, or do they, and I'm just going to go into the dismiss.
In our experience, we're just starting with insect repellent screens in our endophyte collection in wheat and in corn. Yep. And we're seeing repellents rather than insecticidal activity. In the grass system that we use as the exemplar, it's definitely insecticidal, as you've seen from the biomolecule that we've developed as a biopesticide.
Excellent. And I've got a comment here from Carol that I'm just going to read out as well. So one of their most recent projects was isolation, characterization, and identification of endophytes in the roots and leaves of banana plants, sampled from all the different ecological zones in Cameroon and Gabon. We identified different strains of Fusarium, Trichoderma, and Bovaria. Not only did they boost the plant growth, but also some of the Fusarium and Bovaria strains were efficient against banana and plantain and corn borers. So thanks for sharing that with us as well. So you've kicked off a whole lot of discussion, Damien, in the chat. So that's fantastic.
I might just make a comment about that. That's fantastic. I'd really like to chat to you about that, Carol. I think what would be really cool is to go into the native range of banana plants and isolate endophytes from there, because those crops are absolutely soaked in pesticide. So finding bioactive things there is a really good start.
Really interesting. No, excellent. So I hope you two do make contact together, and I can always pass on emails as well, everyone. We've got a question here. Fascinating presentation. Thanks for sharing, from Jan. And can you share a bit about the rice blast work relevant for the ASEAN region? Does this also involve bioprotective endophytes?
Sure. So no. So this is again an endophyte-derived metabolite. So this is a biopesticide developed by Biotelliga. So it's based on essentially a fungus that's usually an endophyte of a range of plants, but it's similar to the NZ7000 story. So it's a sprayable contact by a fungicide, QC'd for the presence of the fungicidal bioactive molecule. It's membrane active fungicide, and yeah, really works very well on rice blast. Also Asian soybean rust, I understand that's becoming less of an issue in Brazil, but yeah, we'd certainly be keen to talk more about that. It's not involving a biological material in this instance.
Excellent. Thank you very much. I'm just going to share my screen while I'm multitasking here, so I may need a bit of help with everyone. Can people see my full screen now, or my notes? Damien, can you see my full screen or my or the notes?
I see your notes. There's a little thing going around.
I know. So there we go. So Damien, just before I go, sorry for that. I just wanted to multitask. Not very good at that at the moment, but Damien, that was really, really interesting. Again, looking forward to coming back to you in a year's time and seeing your progress. And I think that's a good follow up for us. I could have had I think we all could have had just one webinar just on each of these examples, to be honest, because they're so informative and so much to discuss. Your point about working with people in this region is gratefully received, and we'll definitely be reaching out, and I'm sure there's a lot of interest. And we'll also be soon launching the co-design of a big bioprotection centre for Southeast Asia, and that's exactly the type of collaboration that we want to be doing with innovators and entrepreneurs, bio-entrepreneurs, such as all of you today that presented. So thank you so much. And I'm just going to make a very quick summary. So if you can keep with us, I know some people had to leave on the half hour, but I would just like to say it has been a fantastic session. CABI, speaking on behalf of CABI and ASEAN Fall Armyworm Action Plan and CSIRO, we're very pleased to be hosting this session. It was great to hear from Damien around harnessing the biology of plant fungal interactions and how they can contribute to that clean, effective, safe crop protection for the future. Arda, taking the inspiration from plants to design encapsulated biopesticide technology that drives greater performance and efficacy for bioprotection. Fantastic. Neil really showed us a new category of chemical-free sustainable bioprotection through these self-limiting insect technologies. And Neil, just a big shout out there around the importance of communicating these sorts of technologies when you introduce them to farmers and users in the environment. I really liked your focus there. It's a really important part of technology transfer that's often forgotten. So a big shout out there for that. I'd like to thank also all the participants today that joined us. We've had a lot join us, over 160 across the whole session, which is fantastic. And there's always a lot of people that watch the video as well. Leandra, thank you for helping out. Very, very appreciated. Thank you for Roma Gwyn, who also suggested some of the speakers today. So I'm very appreciative of that. And to all of you, I just want to remind you that we have another session on the 25th of August. So we invite you to that session. You can register for all our sessions at the ASEAN Fall Armyworm Action events page. And another shout out, next week we'll be starting quite a detailed session on transboundary pests and climate change and diseases in Southeast Asia with a focus on fall armyworm next week, which will be very interesting. We'll also be looking at some risk assessment across the region there, and then we'll be diving into the genomics, understanding strategies for management in Southeast Asia with focus on fall armyworm on the Tuesday in the following month. So there's lots happening in the ASEAN Fall Armyworm Action Plan. Today, we're very, very privileged to have 3 excellent speakers, and wish good luck in your work ahead, because you've got lots to do, very exciting, very innovative, and we need people like you focusing on new solutions for these emerging and current plant pests and diseases that are really impacting our food systems. So thank you all. Thank you for me. Please keep safe, everyone, and I'll see you next time. Thank you.